Welcome students, I am Dr. James D'Souza, Assistant Professor, St. Xavier's College, Mapsa, Goa. The title of the unit is Preservation and Maintenance of Microbial Culture. Module name, Culture Collection Center. Now this particular unit is from paper Microbiology and Plant Pathology. This is outline. Here concept of Culture Collection Center or culture banks will be discussed. Various types of culture collection center will be explained. Learning outcomes, students will able to explain the concept of culture collection centers or culture banks. Student will able to list the role of culture collection centers. Student will able to describe the types of culture collection centers. Now, what are these culture collection center or culture banks? These are centers that provide authentic organism that can be grown or maintained in laboratory. They are valuable resources for sustainable use of microbial diversity and its conservation. These centers focuses on acquisition, production, preservation and distribution of viable cultures of standard reference microorganism. They play public service role wherein associated information can be obtained and it can be used by future generation. As we know, microorganisms are indispensable for the existence of plant and animal life, and there is need to conserve it. We can't let it destroy. And as India is recognized as one of the hotspot of mega biodiversity, which contain large number of endemic microbial diversity. So, efforts need to be done to tap this immense microbial diversity through bioprospecting projects, which can be exploited for medicinal, industrial, and agricultural purposes. Besides, it takes several years to characterize and develop one microbial culture. And all this work is done at culture centers by experts who maintain these cultures in viable condition. When we are depositing the culture, this is governed by stringent rule and regulation, and intellectual property right of the depositors are protected. Now, what is the role of these culture collection centers or culture banks? They act as a depository for patent cultures, maintain isolated pure culture for extended period in viable state. Now, this is one of the important factor. Next, act as a means of ex situ conservation of microbial resources, serve as a reference for identification and classification of new isolated organism. It provides uniform source of authentic culture to industry as well as academic and all the research institutes. They also develop strategies for those microorganisms which cannot be cultured, preservation and storage of newly isolated strains, then genetically engineered strain with plasmids is also done by these culture collection centers. Basically, this center focuses on basic research in which microbial diversity, microbial taxonomy, microbial genomics and microbial proteomics can be studied. Beside these culture banks, they contain microorganisms which are isolated from various specific environmental niches. Now, depending upon the specialties and function, culture collection centers are classified into three types: private culture collection center, specialized culture collection center, and public culture collection center. Private culture collection center usually maintained by individuals, private laboratories, institutes, hospital and commercial firms. The culture strength here is 20,000 microorganisms. This culture strains is not for public use. Next, we have specialized culture collection. Now, this type of culture collection arises due to taxonomic study of selected specific group of microorganisms. in specialized field. Specialized field include economically important industries such as brewing industry, production of antibiotics, and plant pathology field. Now, one example of specialized culture collection is fungal culture collection of microorganism of University of Philippines. Last, we have public culture collection. It consists of large number of strain which is established for the purpose of public service. Here, number of strain varies from 8,000 to 1800,000. These are some of the major international culture collection center around the world. First, we have NCIB, National Collection of Industrial Bacteria in Britain. Then we have MTCC, Microbial Type Culture Collection in Osaka, Japan. Then we have ATCC, American Type Culture Collection Center, Maryland, USA. Then these are some of the important Indian culture collection center. The first one that is located at Pune, Indian Culture of Industrial Microorganism, National Chemical Laboratory. Second, we have MTCC, Microbial Type Culture Collection and Gene Bank, which is located at Chandigarh. Then we have NCMR, Center for Microbial Resource, again located at Pune. Now, all this major national and international center is governed by International Depository Authority. Currently, India houses 32 microbial resource centers that are registered with World Data Center for Microorganism. Next, we will see why there is need to deposit microbial culture at this center. By depositing microbial culture, we ensure that living microorganism along with their description is maintained. Now, suppose if one researcher is working with a type of a microorganism and he works only with one specific compound from that particular organism. After working with that culture is deposited. After 10 years, 20 years, some other researcher will come. He will look at other compound from the same organism. So, it is useful in this way. Besides this, isolating of microorganism from specific ecological niche, that is specific ecological habitat, can be completed by depositing microbial culture. Here, when you deposit microbial culture, you have to provide information from where it was selected, which is the site, which is the ecological niche. So, once this information is given, the culture is deposited at the culture bank. In future, say after 10 years, 20 years, future researchers can refer to all this information, and if he or she is interested in particular microorganism and particular compound, they can go for collection. But one cannot be assured that same strain will be isolated from same ecological site. Coming to summary, in this topic we have discussed culture preservation and maintenance, how it is done in culture collection banks or culture collection center. Then how culture collection centers or culture banks are classified based on specific function, okay, and how these culture collection centers or culture banks are utilized by scientific community to do advanced studies. These are references. The first one, practical microbiology book. Then there is one research paper which was published in 2014. Thank you very much.